Well, hey again, folks. Mr. Bleecker here. In this lesson, we're going to follow up on what we had been studying with osmosis and diffusion, and we need to take a closer look at how water flows. Now, we'll focus on osmosis more in this lecture, and it's because water is so important to life, we want to know how it flows. What is its motivation to flow into or out of a cell? Or is it in equal amounts and just at equilibrium? There's three terms that you're going to need to know once this lesson is finished, and they're hypertonic, hypotonic, and isotonic. Now, the term tonicity really just means how concentrated is the external environment compared to the internal environment. And when we're talking about concentration, we're talking about solute concentration. By solute, you could think of things that are dissolved in solution. That could be sugar, that could be salt, those could be proteins. But these are things that water molecules, being polar, are attracted to. So we want to focus in this lecture series about that net flow of water. So we are looking at osmotic flow and osmotic flow motivation. Okay, so I've got a neat little thing to show you here. Here it is. Now this is fun. You've probably done this in elementary school, where you take an egg, and in this case you can use tap water, but you get really great results if you use distilled water or filtered water. But tap water will do. And in beaker A, you take an egg, and you have, for about 24 hours, you have to dissolve the shell. So you get to have a little fun with vinegar. 24 hours in advance, you use vinegar, and vinegar removes the calcium carbonate shell. Now, that leaves just the membrane and then the egg inside. So once you've denuded the egg or removed the shell, you can put the egg, well, you get a couple of them ready to go, into different solutions. And in this case, in A, they're just using tap water, but you could use distilled water, as I said. And in B, you put the egg minus its shell, and all it is really, after the shell is gone, it's just a membrane. You put that into corn syrup, like this. Now, there's some great time-lapse sort of animations that I've put on the course site, but I'll just describe to you more or less what happens. When you put an egg into this solution, outside of it, the solution's very watery. There's nothing really in it. And the most concentrated thing between the egg and its environment, what really is the egg. Inside are amino acids and sugars and salts and things like that. So the egg is a very concentrated thing in this environment. And you've surrounded it with dilute tap water. There's very little in tap water. It's just water. In this case, you've placed the egg into a really concentrated, syrupy environment. And one thing that I wanted to get you to understand about water is, as a polar molecule, it's going to go towards the solute. It's going to go towards the protein particles or the salt particles or towards sugar. It's going to want to make an association with them. So if water basically wants to go to the, well, where the action is, where the solute is most concentrated, then what you have to do is sort of think about, well, what is water's motivation? What's its osmotic motivation, or which way is it going to flow? Well, in this case, give it about 24 hours, we can see that this egg has swelled considerably, taken on a lot of water. So water has wanted to flow inside of the egg. The water outside the egg is able to pass through the membrane, and it wants to go where the action is, where the solute is. So the egg in this case was the most concentrated thing in the region. Now, if we look at scenario B, the most concentrated thing inside of this beaker, as far as water is concerned, is the environment. So water flowed through this membrane and into this corn syrup. And when you're done, you can actually, the corn syrup, it becomes very liquidy, and it flows much more easily, much less viscous. So this experiment is fun, and we'll do it. It's designed to prove the difference between a hypertonic and hypotonic environment. This environment right here, outside the egg, is very hypotonic. the environment, what the egg's residing in. The environment outside of this egg is very concentrated. We say its tonicity is high, so we say it's hypertonic. And we'll get to isotonic later, but if you place something in this egg, for example, into a hypertonic environment, the net osmotic flow is out of the egg. But if the egg is the more concentrated environment, the net osmotic flow will be into the egg as opposed to out of the egg here. Okay, great experiment, lots of fun. Now, to help you visualize what's happening here, we have an animation. So these little cubes here just represent solute in the environment. We could call it sodium chloride. It could be sugar. And if the concentration of these solutes are equivalent, then really we're kind of at equilibrium. So the direction of the flow of water is going to be more or less even. So the amount of water flowing in is sort of equal to the amount of water flowing out. And you'll notice, let's pause that the cell membrane remains evenly inflated, if you will, because it's losing as much water as it's gaining. So if we have an even match here, we say that's isotonic. Iso meaning same, tonic meaning concentration. So the concentration inside and outside of the cells is about even. And this is good. It's at equilibrium. This is a good scenario for the cell. Now, if we look at a scenario here where the environment is more concentrated, and here's a great example. You've been eating salt and vinegar chips for the better part of half an hour, and you have made your extracellular fluid really, really salty. What will happen is you'll see there's sort of a mismatch here. The environment is more concentrated than the inside of the cell. Okay, so now we have to do a comparison. There is less solute inside the cell than there is outside. Okay, so you have to think about this. That means that this is more of a watery environment. This is less concentrated. Now, if you're a water particle, you're going to go where the action is. This is very similar to the egg being placed in corn syrup. So when I play this animation, you'll see that the water particles will flow out. And it gets to a point right here. Ah, shoot, I can't pause it. Let's see if I can hit pause right there. Boom. Now that the water particles have flowed out, this sort of is an attempt to dilute the environment. Water particles, the net osmotic flow is out of the cell. Now, as the cell loses its water, what happens is it loses its internal water pressure. This happens to plants when they become, they get dried out over time if you forget to water them, for example, and they wilt. You see this sort of wavy appearance of the membrane here. We say that this is sort of a scalloped appearance, and the name for this is a crenated cell. Crenated is a term that means scalloped or wavy edged. So what can happen here is our cells start to experience a net loss of water, and as the solute concentration in our blood rises, our brain becomes aware of the concentration of the blood, and that will promote thirst to try to even out sort of the osmotic flow. Third scenario for you here, and we'll look at this one. This is one in which when we place the egg into the purified water. Let's say it's in distilled water. In distilled water, there are very few solutes. One of the whole reasons of distilling water is we evaporate it and we leave behind the impurities, and that's actually a great way to sterilize water. Now, in this case, we could say this might be a bit more like tap water. There's still a few solutes in solution, but it's pretty dilute out here. So you'll notice this says the water concentration's high and the solute concentration's low. That's what these little brackets mean. It means concentration of. So low concentration of solute, high concentration of water, blah, blah, blah. Really what that means is the cell here is the most concentrated act in town right now. Water is going to want to flow in to sort of dilute this high concentration environment. There's more attractive things in here, if you will. So if we play this now, and I'll just pause that. Oh, too fast. Right there. Okay. Now the net osmotic flow was into the cell. So the cell, in this case, is the most hypertonic thing that's basically in this demonstration. If the cell is relative hypertonic, then the environment is relatively hypotonic. What does that mean? It means water is going to flow into the hypertonic environment. There's more solute here. It's more concentrated. So water will flow in there. Interesting. Interesting. So what we've got is we've had a chance to look at isotonic environments where it's relatively the same. We've had a chance to look at hypertonic environments where the net osmotic flow in this case is out of the cell. And we had a chance to look at an environment in which the environment is hypotonic, so there's a flow of water into the cell. You have to think about which one is hypertonic compared to the other one. Really, the trick is, what's the most concentrated thing inside the cell or outside of the cell? And you have to designate one as being hypertonic or hypotonic. And if they're the same, you say it's an isotonic situation. Not bad. Now, I'll just zoom in on this a little bit. This is sort of a visual representation of what had happened. And you can look at the flow of water in this case, and you could say, well, what's the osmotic flow? And it really depends. It really depends. You just have to say, is it in a dilute solution or a concentrated solution? Now, you can have a little bit of fun with this. You don't have to say goodbye to your vegetables. And in fact, if you go to a restaurant, vegetables that have wilted don't have to be thrown away. All they've done is, well, wilted. So what's happened in this case is your vegetable has become dehydrated. Fancy term for wilting. Now, you can give lettuce, for example, a cold water bath. You really don't have to put it in the fridge or anything like that. You just have to give the lettuce a chance to regain the water that it has lost. And it doesn't take very long. In restaurants, you'll see that celery sticks are placed in sort of upright cups of water. And that's kind of important because we don't have to lose our vegetables, right? They can be restored just by net osmotic flow. Because if you place this in water, the vegetable itself would be the most concentrated thing, and you'll have water flow back into it. It's pretty cool. So what happens with vegetables when they lose their, basically when they lose water, is we say that there's a net flow of water out. So just pause this. So as your plant is left for a long period of time, and here's the cell wall around the outside, right? And then the cell membrane on the inside, you could see the cell membrane starting to come in a little bit because this plant is losing water. It's using up water as it's doing photosynthesis. This white thing in the middle is the big old water vacuole, the large reservoir of water that plant cells have inside of themselves, and that nucleus over here. So if plants need water for photosynthesis, they will use up this store and then they begin to wilt. So you can see that vacuole begins to lose water, and even it loses water, and there's not as much pressure inside the cell to hold the cell membrane up against the cell wall. So this is wilting. The cell wall itself will sort of, well, it doesn't collapse, but it becomes a little bit, it doesn't have as much pressure against it, so it folds in a little bit, and you get a wilting process like that. And the plant is fully wilted. Now there's a term for this. This is called plasmolysis, where there's a net loss of water from a cell. Plasmolysis. Know that term. The good news is this is completely reversible. When you water a plant, it goes back to this. And then we just repeat this process as we typically forget to water our plants again and again and again. But the good news is plants can be rehydrated, so don't throw them out. What it's called, really, it's called Turgor pressure. And when a plant loses water, we say it's been plasmolyzed or plasmolysis. If a plant, for example, plants floating in the ocean, they can take on water as typically is what they need. So they'll be in an environment where they could stay hydrated. So they're fine, right? That's an isotonic state. In a hypotonic state, there is a net inflow of water. So if you take a plant and you water the daylights out of it, it will simply just rehydrate. This becomes a little more interesting if we get away from plant cells, and we talk a little bit about animal cells. And one of the best ones to look at is, red blood cells. Now you take red blood cells, and typically in our bodies, if we're well hydrated, it's an even sort of game. We're isotonic. So the net flow of water in is approximately the same as the net flow of water out. That's great. Now, this is why you do not drink distilled water. The people have killed themselves drinking way too much purified water, because what that does is that waters down the blood to a point where the environment for the red blood cells is extremely dilute. And we know what happens. If the environment is super dilute, then the red blood cell is the most concentrated thing. So what happens is there's a net flow of water in, and you'll see that the cells begin to swell. And remember, there's no cell wall around animal cells. And if you give them too much water, they'll simply burst. And that's a fatal condition. You can't drink too much purified water. Now, if you look at another situation, let's say you're wandering in the desert, you've been lost, you got cut off from your tour, and your blood becomes super concentrated because you can't get enough to drink. Well, what will happen is the concentration of solids and waste products around your cells becomes really high, and you don't have very much water outside of the cells. Well, the cells, kind of like the plant we just looked at, will begin to lose water. The environment is so concentrated that the net flow of water is directly out of the cells. So the cells are in a hypertonic environment, and they become crenated here, those little scalloped edges. So this is where you want to be. This is homeostasis right here. But we get into situations where we don't get enough to drink, right? And this is obviously devastating for the cells. They can die under these conditions. This condition here, where they're surrounded by too much water, right? And then you get, now this right here, we say that this is cellular lysis. This is called cytolysis. Cyto meaning cell and lysis meaning burst. So cytolysis for cells bursting. But I wanted to show you what that looked like. Here's what the blood cells look like when we look at them with electron micrographs. Ooh, I'll just go back one. I'll just zoom in. So on the left, you could see the crenated cells as they undergo plasmolysis. Now, plasmolysis is when cells lose water. We saw that when plants basically wilt. And over here, in a hypotonic environment, believe it or not, these are red blood cells. They're supposed to look like donuts from the side, but they're swelling up like a balloon that's got too much water in it. And again, we're looking at about a 20 micrometer scale. What you should be seeing is this. You should be seeing the red blood cells sort of looking almost like, well, hollow in the middle, right? Over here, you can see some that still appear normal, but in this case, they should look like that. Okay, if it's isotonic and they're gaining and losing water in an even rate. Now in this video, I want you to keep, it's kind of interesting, I want you to keep your eye on this red blood cell here. This red blood cell is the most concentrated thing in this region. This is purified distilled water. Now watch what happens to this red blood cell as it takes on water. You see it's starting to it's taking on water, it's starting to swell, and keep your eye on the cell membrane, especially right there. Boom. That's called plasmolysis, folks. And what has happened, or sorry, not plasmolysis, that is cytolysis. The cell has taken on so much water that the integrity of the membrane gave way, and cytolysis, correct myself again, cytolysis has occurred. So I'll just jump out there. Sorry. Okay. So basically what I wanted to get you to get out of this lecture was the differences between hypertonic, hypotonic, and isotonic environments. There are some notes that I'll give you for this section, but really we'll just treat it like a handout so that you can have the material for this relative sort of lecture series. But again, that's it, folks. Thanks for listening. This was a little lesson about tonicity, and thanks for popping by, and have a good one.